I will start the first session by introducing the moderator of the first session. The moderator of the first session is Marlene Camacho Rivera. She is a doctor, and she also has a Master's of Science and also a Master's in Public Health. She is the Assistant Dean for Student Affairs and Assistant Professor in the Department of Community Health Sciences at the School of Public Health at Downstate. So she's going to be introducing the speakers for the first session of the symposium. So Marlene, can you come to the podium? Thank you.
Good morning, everybody. Good morning. Have the pleasure of moderating our first session, which is going to focus on colorectal cancer. And our first presenter will be speaking virtually, Melissa Kramer. Melissa has a Bachelor of Science degree from the College of William and Mary and is a DNA sequencing technician at Cold Spring Harbor Laboratory, one of our partners in this symposium, and is also a scientific data analyst in the Macombi lab at Cold Spring Harbor. And Melissa today will be presenting on increasing representation of African Americans in the assessment of genetic contributions of colorectal cancer. Thank you, Melissa.
Thank you. Good morning. Share my presentation. Good morning. Thank you for the opportunity to share this work, which is a collaboration between Cold Spring Harbor, Downstate, Northwell Health, and the New York Genome Center. So racial disparity, of course, has been studied across cancers and in colorectal cancer in particular. And colorectal cancer is one of the top 10 diagnosed cancers in the United States. It's been found that African Americans have higher incidence and higher mortality than people of European descent. And though socioeconomic factors and access to care do impact this, it doesn't fully explain the disparity. So population genetic differences and environmental exposures are also thought to contribute. And in terms of treatment, these genetic profiles are particularly important, and a recent MSKCC study found that African Americans are less likely to have actionable mutations that will respond to current therapies such as immunotherapies or drugs that target BRAF mutations, which are less prevalent in African Americans. So we employed two cohorts of colorectal tumor samples for this study. The first was derived from FFPE samples, 52 African American and 51 European American tumors, on which we performed exome capture and sequencing. These samples did not have matched normals, so we employed bioinformatic pipelines to try to prioritize and stratify somatic versus germline mutations by using sequencing controls, normal controls, as well as allelic fraction filters. As part of this study, we also sequenced some polyp samples. These were not matched to the same individuals that the tumors came from, but a separate set of polyps to look at pre-neoplastic lesions and transitions from adenomas to carcinomas. These polyp samples actually included both tubular adenomas and serrated polyps. The second cohort was derived from the Polyethnic 1000 Consortium. This consisted of 42 African American tumor samples and 10 European American tumor samples. These had tumor normal comparisons. There was normal adjacent available for these samples, which help with the variant stratifications. And these were fresh frozen samples as compared to the FFPE sections. These were whole genome sequenced rather than just exome capture sequencing. So there are some technical differences in the two cohorts, and we'll make note of that as we go along. The P1000 Consortium also had a subset of tumors that were transcriptome sequencing profiles were derived from RNA-seq. These were tumor only, and they were a subset of the same tumors that we did genomic analysis on. For the study, we employed genetic ancestry rather than using self-reported race in assigning our cohorts. We compared the variant frequencies of our samples to the 1,000 genome superpopulations, and we required a cutoff of at least 70% of either African American or European American ancestry to assign individuals to our cohorts. to control for the admixture. So we did see in these two cohorts, the left panel here is the whole genome sequencing cohort, in which we had more individuals of African American origins, denoted by the orange bars here, compared to the blue bars, which indicate the European American samples, and the pink is Asian American. We then looked at our exome capture samples from the FFPE sections. Here we had a more even distribution of African American and European American samples, denoted again by the orange for African and blue for the European. And we also looked at the subpopulation, subcontinental ancestry of these samples. We found that for our African data sets, very similar to TCGA cohorts, we mostly had West African subcontinental ancestries. And for the Europeans, it was predominated by Italian or British. So we first wanted to look at the tumor polyp comparisons to look at changes that are associated with transitions from pre-neoplastic to carcinoma tissues. And so we compared here with these forest plots the African American tumor and polyp samples and the European American tumor polyp samples. Again, noting that these are not from the same individuals, but separate samples. The polyp samples, again, were comprised of both tubular adenomas and serrated polyps. And what we found when we looked at these forest plots, the blue bars indicate genes that are more highly mutated in the tumor samples, and the red are more highly mutated in the polyps. What we found here is that, in general, for both African and European tumors, APC mutations are early mutations, so they tend to be enriched in the polyps. Whereas TP53, KRAS, and PIK3CA seem to be more enriched in the tumors and are later driver mutations. But one thing that we noted specifically in the European cohort was that the serrated European polyps showed BRAF mutations as the early driver, rather than more of the APC mutations. So you can see that there's an enrichment here for BRAF. This was specifically driven by those serrated polyps. We then employed a pipeline back to our tumor samples to prioritize driver mutations. So we specifically targeted loss of function mutations and high impact mutations, such as missense mutations that would be noted as pathogenic in ClinVar, or cosmic, or seen in the TCGA cohorts, to really prioritize more likely drivers. And what you'll see here in this panel on the right is an oncoplot of the most highly mutated genes across, this is the whole genome sequenced P1000 cohort. And you'll note right away that some of the samples seem to have much higher tumor burden than the others. And indeed, those samples turned out to be MSI-high samples, so they had microsatellite instability. And they do show different variant profiles than the microsatellite stable tumors. So for further cohort analyses, we did separate out the MSI-high samples and focus on the microsatellite stable for the cohort comparisons, which have more likely tumor burden. We then summarized the mutations in each of these cohorts, so both the whole genome sequencing cohort and the exome sequenced cohorts. First, for the whole genome sequencing, again, focusing on the microsatellite stable tumors, we looked at the variant classifications. They were dominated by missense mutations with similar numbers of frameshift and nonsense mutations as the second and third most common. And they were also dominated by C to T transitions, which have been noted in colorectal cancers, indicative of 5-methyl-C deamination. We looked at the top mutated genes in each of the cohorts. And in this case, because we had much fewer European samples, the top genes are a bit different than in most of the other cohort comparisons that we've done. That's just a factor of sampling. But again, we see, as expected, the top drivers in colorectal cancers, TP53, KRAS, PIK3CA, APC, in these different groups as the top mutated genes. We then compared that to the whole exome sequence tumors, very similar profiles in terms of the mutations, somatic mutations. And again, top genes, APC, KRAS, P53. And what you'll notice is in most of the cohorts, when you're comparing similar numbers of European and African samples, there seem to be a very similar frequency of mutations. So for example, for P53, in these cohorts, again, the whole exome sequence cohorts had approximately 50 samples each, well matched, and the frequencies are quite similar. But what we did notice was that we were seeing a trend for KRAS to be more enriched in the African-American cohorts than in the European cohorts. And of course, this has been seen in multiple previous studies, that KRAS is more prevalent in African-American cohorts. We've seen it not at a statistically significant level, excuse me, but as a trend in frequency across our cohorts, which are small at this point. And so you can see that in all of the African-American sample sets that we sample here, it does seem to be increased frequency for KRAS. And then we expanded to some other European data sets, some larger data sets for comparison. Both the TCGA colorectal cohort, African-American and European Americans, and the samples we had downloaded from a Dana-Farber study of colorectal cancers. These were exome sequences, tumor normal matched. And so again, when we look at KRAS mutation frequencies in these larger cohorts for comparison, it does again seem like the African-American cohorts have increased frequency across the board here. And of course, these mutations include the known activating mutations that have been seen to be enriched in other studies. We also employed this pipeline to try to look at germline variants. This, of course, is more challenging. There are many germline variant differences, and we tried to prioritize differences that look like they had a high impact. So the same type of prioritization that was done on the earlier samples for loss of function or high impact annotated variants in ClinVar. And what we found was that we did find rare mutations in this gene AHNAK2 that did seem to have a trend, again, not significant, but a higher frequency in the cohorts that we sampled for African samples compared to European. And this gene has been known to be differentially expressed in colorectal cancers that have KRAS mutations. And high expression of this gene has been linked to poor survival in both colorectal and pancreatic cancers. So if this could be a background germline mutation that might be more enriched, could contribute to the disease in African Americans. And there are also somatic mutations noted in this gene. Again, looking at the cohort comparison of mutational frequencies, this forest plot shows the blue bars pointing toward genes that are more enriched in our African American whole genome sequencing cohort, which is 33 samples. And the red arrows on the left indicate genes that are more mutated in the European samples from the TCGA COAD. And again, we stratified these for microsatellite stable. And what we found is, though our numbers are small here for the enrichment of the genes in the African cohort, there are some interesting genes that come up as suggestive of enrichment. ZNF469, NAA15, ZBTB7A. These have previously been linked to poor prognosis and increased tumor progression in colorectal cancers. SMAD2, of course, is a known tumor suppressor in colorectal cancer as well. And though I don't have the forest plot here, we also did this analysis in the whole exome cohorts. And there we saw some indication, again, of genes with a slight enrichment, smaller numbers, of genes that had been previously associated with colorectal cancer. Tumor suppressor genes were more highly mutated. FBLM1, which is hypermethylated in colorectal cancer, also seemed to be more mutated in the African samples compared to European. But what really interested us when we were looking at this was in the whole genome sequencing cohort, it looked like APC was actually depleted in the African American samples compared to the European samples from TCGA, which is the top gene in the figure here. We then looked at the Dana-Farber samples, again, another larger set of European colorectal tumors. And here again, although APC did not reach the significance of making the cutoff for this plot, when we look at the frequencies within the cohort, again, we do see that the African cohort does seem to have lower, for about 45% frequency than the Dana-Farber cohort, where there are about 67% of samples have an APC mutation. And again, the genes that were enriched in our African cohort compared to TCGA came up again as enriched compared to the European samples from Dana-Farber. So some concordance there, again, with small numbers. APC mutations have been studied, of course, extensively in colorectal cancer. And though some larger studies have found that African Americans have more APC mutations, including the Foundation Medicine study from recent work, other studies have actually found that APC mutations act differently in certain African American cohorts. So an MSKCC presentation at AACR in 2023 found that APC mutations in African Americans were not predictive of outcome the same way that they were in European or other ancestries. So typically in European or other ancestries, they would correlate with a better outcome, but the APC mutations in the African tumors did not seem to improve survival. Also, several studies have shown that tumors without APC mutations have worse prognosis and earlier onset than tumors that have one or two truncating mutations. And interestingly, similar to our study, a recent study showed that in the West African Nigerian population, there were reduced APC mutations and increased KRAS, very similar to what we're finding in this cohort, compared to the MSKCC European samples. So to further explore this, we looked at mutations in the oncogenic pathways across each of our cohorts. So comparing, again, our smaller African cohort to the European TCGA and Dana-Farber sample sets to see if not only APC, but again, the Wnt pathway in general had a difference across these cohorts. And in fact, we do see that it looks like there are fewer Wnt pathway mutations. Approximately 66% of the samples had a Wnt pathway mutation in our African cohort, compared to 88 or 90% in the European cohorts. We then looked to see if there were other hits to canonical genes in the Wnt pathway that could be affecting the African American samples. We did not see significant hits in CTNNB1 or RNF43, or even SOX9, which is known to interact with the Wnt pathway. Of course, there could be other mutations, either in genes that interact with that pathway, or things such as methylation, which we did not assess in this study, which could cause hits on genes in those pathways that would affect differently in the African Americans. But again, something to delve into. We then looked at somatic interactions in the cohort. And again, we noticed for our P1000 African cohort compared to the TCGA European cohort, excuse me, that although some of the variants seem to be acting similarly, for example, PIK3CA seems to be mutually exclusive with TP53 in both cohorts. And PIK3CA also seems to be interacting as a co-occurrence with KRAS in both cohorts. When we look at APC, we actually saw a difference between the way that it's behaving in terms of interactions with P53. So in the TCGA cohort, it does seem to have a co-occurrence with APC and TP53, whereas in our African cohort, it seems to trend more towards exclusivity. So not exactly the same interactions. We then investigated the local ancestry of these whole genome samples. In the samples that had no APC mutation, we looked across the APC gene and found that there are West African associated haplotype blocks, including this block here, block 6, which seems to be more frequent in samples that do not have an APC mutation compared to those that do have APC mutations, such as the SNPs shown here that were mutated in our cohort. And so we want to really follow this up with the exome samples to see if this pattern continues. We did not really see a depletion of the APC from that cohort in African versus European, but of course we have samples with both APC mutations and no mutation. And so we want to see if this haplotype block continues to predict APC status. So recently, very large studies have been done of mostly European individuals. The two studies I have listed here have over 1,000 or over 2,000 individuals of mostly European descent. And they note that there are drivers that they're finding in colorectal cancers. Some of them are genes that either have not been associated previously with colorectal cancer or in any cancer in general that are being shown as recurrent mutations in colorectal tumors in the large data sets. And what we found is this is just an oncoprint showing that when I look at the gene list from these studies, we actually do see a significant proportion of the genes that they noted as drivers, even within our small cohort. And this includes some of the genes that were not previously associated with colorectal cancer. So I think it is really important to expand the numbers of African Americans that are sequenced so that we can understand the genetic variations that exist and more of the recurrent mutations for these individuals. We then looked at mutational signatures, so changes that happen in the tumor across the genome for each of these whole genome sequence samples. We fit our mutational signatures from our cohort to those from the COSMIC mutational signatures database, and looked at the proportion of each of the signatures across both the European and African samples. So the main signatures that we saw here were signatures that have been associated with colorectal cancer. Signatures associated with aging, our cohort is mostly over 60, deamination of 5-methyl-C, defective DNA mismatch repair, and polymerase deficiencies. Again, these have all been noted to be prevalent in colorectal cancers. And what we'd like to do is really try to expand the comparison of the proportions that we're seeing within our African cohort to a larger European data set and see if maybe there are some differences in the signature proportions. We then looked at the RNA-seq, again, so this is transcriptome sequencing of a subset of the tumors from the whole genome sequencing. And we wanted to compare to TCGA Europeans because we have only a few, approximately 7 European tumor samples that were sequenced as part of this project. And we are going to refine this analysis to include covariates for CMS subtyping to ensure that differences that we see when we are trying to compare ancestry and not an effect of the CMS subtyping, which I'll explain more later. And what we did find with the differentially expressed genes here in our African cohort compared to the TCGA Europeans, when we look at KEGG pathways for the genes that are differentially expressed, we find pathways involved in immunity and inflammation are affected in the African American samples, as well as Wnt signaling, and this PPAR signaling pathway, which is involved in lipid metabolism, but also in cell proliferation. And so these could also affect disease in these African American samples. We then did consensus molecular subtyping to stratify the tumors in the cohort. Again, these are subtypes derived from expression profiles from different gene sets and pathways. CMS1 is typically associated with immune response. And again, our MSI high tumors, of course, did stratify into the CMS1 subtype. CMS2 and 4 are considered more aggressive subtypes. And we did find that a high proportion of our African American samples fell into the CMS2 and 4 categories, with a small proportion in CMS3. And we were interested in this because though we have a small number of European samples at first look, we thought that the African samples might be more enriched in the more aggressive subtypes. However, when we went to look at TCGA, we don't really see a significant difference in the subtype assignment by ancestry. So although in our cohort, those more aggressive subtypes might inform that these samples may have a more difficult treatment response, we would need larger numbers for comparison, I think, to try to see if there was an association of any enrichment by subtype, by ancestry. We then looked to see if we could correlate CNV profiles with those CMS subtypes. Again, knowing that CMS2 and 4 have higher chromosomal instability and more amplifications and deletions across tumors. And again, we were able to stratify these tumors by the CMS subtype using the copy number profiles, which could underlie the gene expression changes that stratify the subtypes. And so what we found was that CMS1 is mostly flat profile, indicative of the MSI high tumors, and CMS2 and 4 show amplifications of chromosome A, which is MYC, as well as chromosomes 13 and chromosome 20, which have been associated with cancer progression. And then the subtypes of CMS2 and 4 also show 17p loss and 18q loss, which larger studies have also confirmed are indicative in many colorectal cancers and quite prevalent deletion profiles. So again, this is another way that the samples can be characterized and stratified. We did look at structural variants. They were called across all of these samples, and the majority of samples in both the African American and European cohorts had a predicted alteration that would at least overlap part of a tier 1 cosmic cancer census gene. And so even if these were intronic or regulatory regions of partial overlaps, we'd like to look at this effect on expression in the cohort, because we know that SVs can have a regulatory impact on gene expression for these genes that could impact the cancers. And finally, we looked at microbiome analysis. 20 of the African American tumors from this cohort and 20 non-tumor tissues were selected to undergo 16S sequencing to look at microbial content. And what was found was that signals were higher for fusobacterium in right-sided tumors in the African samples. And those fusobacterium positive tumors had abundant oral pathogens and fewer probiotic beneficial bacteria in the gut. And when a GO enrichment was performed from the RNA sequencing of those same tumors, it showed immune activation and inflammatory pathway activation. And so, interestingly, African Americans have a higher incidence of right-sided tumors, and they tend to not respond as well to treatment. And so this microbiome analysis could inform that as well. For ongoing work, we are still looking at the non-coding variant analysis. Again, much like the structural variant analysis, this can inform regulatory impact and expression differences. We want to continue the work with comparing the mutational signatures to a larger cohort, refine the transcriptome analyses, and in general, as a larger note, of course, we know that long-read sequencing has been shown in many studies to uncover additional variants that could be missed by short-read sequencing. And so that could open up avenues of variants that impact disease as well. So I'd like to thank everyone who's involved in the study, the Macombi lab, Krasnitz lab, Bayaz lab, and Tuvison labs at Cold Spring Harbor, Jeff Boyd at Northwell Health, as long as Nyasha Chambwe's group, Laura Martello-Rooney at Downstate, and Dr. Zhang, as well as Dr. Rishi for pathology, and of course the New York Genome Center and Nicholas Robin's group. And with that, I thank you and take questions.
Thank you. We will be taking questions after our last presentation within this particular panel. So please hold your questions for Melissa's presentation until the end. Thank you, Melissa.